Thank you. Good morning. So we're going to listen to Hernán on stress management for extensive crops. After that, we're going to have 10 minutes for questions. Hernán is the technical manager at Seed Care and Biologicals. Hernán, the floor is yours.
Hola. Bueno, buen día.
Good morning. So today I'm supposed to talk about this topic. Thank you, Pablo, for the introduction. So this topic is very current, this topic of the abiotic stress. We know in the last seasons, we know what we've experienced and its impact on productivity. So the idea is to go deeper into this during my presentation about the effects of the abiotic stress on crops and how we can manage it. And what is the proposal from Syngenta that we're working on in terms of products and technology, precisely to mitigate this issue? First of all, something that is worth explaining, stress has multiple causes. Anything that breaks down with the homeostasis, that chemical balance within a cell, is a cause of stress. And this is not an exceptional condition, because what is usually thought and what we have in our mindset from the point of view of the advisors and the farmers is, well, in this season we had a stress condition, or in a certain period we had a stress condition, and it's just the opposite, or it's a little different, because we have to learn to coexist with stress in order to manage it throughout the crop cycle. But fortunately, there are tools that help us do that. It requires our intervention, of course. To give you some figures, if you look in the global literature, there's a lot of material about stress. And if we try to weigh the losses, the average global loss, well, abiotic stress is precisely for many reasons that we are going to review. Some are more important than others. They can even cause losses of 70% of the yield of the crops that we can reach, which is a lot that we are putting at stake just in the abiotic side. Buchanan work helps us weigh that. And going back to the factors, of course, we know very well, I'm sure there will be a lot of talks about this, we know very well the biotic problems that we can face, or the biotic adversities like grasses, pathogens, weeds, etc. Now, in the abiotic side, it requires the study of dynamic factors, as simple as water, radiation, temperature. I mean, everything we need as inputs for productivity. If it reaches a state of imbalance, because these are dynamic factors in time, they are causes of stress. So I just mentioned something. The plant naturally has a balance physiologically. I mean, when the conditions are normal, because there's no deficit or surplus of water or of temperature, the balance is maintained, which is called homeostasis. It's like the girl you see there in the bottom left that is trying to put down the crop, but it goes back again. It defends itself. When the conditions are normal, then understand that homeostasis is achieved, and the productivity depends on the genetics, of course, but then you have to manage the biotic factors, that is the other big group of adversities. But we should understand that homeostasis is something we have to achieve besides combating the biotic issues. This is important as a concept. What about the response of the crops to the stress? Of course, I included this here in this scheme because the response is very complex from biochemical perspective and the molecular perspective. There you see a terrible map that is unintelligible of the metabolic pathways, and it's actually not complete. It's a snapshot. It's just a piece of all the things that are there. That is, the stress is something that is also involved, or these factors that generate stress actually are involved in a lot of metabolic pathways, stopping the cell. Now, at the molecular level, this is manifested in something that we then see in the field, and that is basically that the plants perish because the cells die, and the cells in general, the cause of the stress, are dehydrated. Why is that? Why do we see it that way? Because at the molecular level within the cells, there are molecules that are a common factor, or pieces of molecules that have an electronic load that breaks the cell membranes. That's a common factor that we can have. Hydric stress, we can have thermal stress, but there's always a manifestation of oxidant aspects. That's a molecular side that works as a dynamic driver of our condition, because anyway, the oxidant radicals are produced within the cells. That's why we're going to see that even if we have causes that can generate stress, there may be a balance, as we see in the triangle in the top right, because there are molecules that also help us within the cells to manage that level of oxidant radicals that are permanently attacking the membranes, the chloroplasts, and when that is in an imbalance because the production of antioxidants is low, or the oxidant radicals are in a large amount or a large concentration, actually, there we have the condition of an oxidative stress at the cellular level that, of course, has a series of consequences, maybe molecular damage, if it's light, or as we move forward, we destroy the cells, it ends up in a cell death and then in a tissue death. And something as essential as photosynthesis is affected. There's a measure of productivity or that is related to the growth rate, actually, that is the photosynthetic efficiency. If we have a stress condition, even the photosynthesis is affected. And what we know that has one of the parts, I mean, this photosynthesis that happens like this, just when we are about to collect the energy we need for the cell through sugars and what we've seen in the previous formula, along the way, we have these oxidant radicals. I mean, something as essential as photosynthesis is affected. It becomes like a weapon against the plant itself. So for that, fortunately, we have tools that allow us to rationalize these processes, to try and maintain that normal condition. That's the big group of biostimulants. And here I just wanted to show you, because there's it's a whole world of elements, but basically what we have to think about is what's the role, what's the functionality of the products, and what do they deliver? And you can see here that the focus, basically, as we said, the stress condition is quite wide in terms of time in the crop cycle, and therefore we should try and protect the plants from very early stages. Of course, this is aligned with the natural cycle. If we go back to our time in college, we know that every 15 days there are hormonal cycles that mark the pace to move through the different stages: emergence, growth, vegetative stages, development. Those hormone levels that happen every 15 days, it's like a lab report to see how we are doing. And those hormones end up triggering all the pathways that we saw before in that scheme that was impossible to read. Within the large group of biostimulants, we have the bioactivators. These products have a diverse profile in terms of their functionality. And now we're going to go a little deeper into this. But the purpose of the product is in the abiotic stress, we should be able to move the crop to the normal condition, the homeostasis, what we are looking for, and in the normal condition, maintain and increase productivity. That is where I give you this proposal by Syngenta that we've been working on for several years now, that is Megafol. Megafol is a product that is applied on the leaves, but it's a formulation obtained from natural ingredients. Now we're going to see how we work with them, that come from algae, basically, extracts. And they have a particular technology, which is a process technology, basically. The brand is Gia Power. For some years now, we've been working at Syngenta with, since a merger with a company called Barbero. Perhaps you've heard of it. Now, Syngenta Biologicals has a part of Barbero, and we've gradually incorporated the portfolio and the designs, and now we are designing products within Syngenta Biologicals that have this technology. And now, before explaining Megafol in more detail, I would like to tell you more about this because it's quite special. What is the logic when you develop a biostimulant product? Well, to reach that homeostasis, I need hormones, nutrients, etc. So I use a given quantity, and with that quantity, I reach the level of sufficiency or homeostasis. That may sound rational, but in practice, it's difficult to achieve. So what's different about the Gia Power technology? That beyond the content, it focuses on the functionality that those components need. We are independent from how many micro or macronutrients we add to the formula, or how much vitamin I have, or how much hormone I have within the bottle, to put it literally, and I focus instead on the effect of those components that I have. So it's a paradigm shift. Of course, when we speak of algae extract, there are known extracts But keep in mind, to understand this technology, that it's not that as I have a given amount of alginate, etc., that will promote the growth of this part of the crop. On the contrary, it's a formulation that, based on how the prototypes are made, that is one of the subsequent stages. Once you choose the extracts that will help us, some are known, others are not so well known, to have an anti-stress effect over the growth. This is all done in Italy. There's a prototyping of a lot of potential formulations. But of those hundreds of formulations, the most important stage comes in this filter, the biological triage. That is, understanding the effect, and particularly based on genomic technologies, of those components that I have at the level of the genome of the crop. What are they generating? Are they activating? Are they silencing genes? At the level of the phenomics. As an initial overview, I apply an extract of that. How much does it allow me to grow in the roots, in the leaves? What does it deliver? And then, with all those components, based on those results, then the genomic part is done with microarray DNA technology. I'm not a molecular biologist, but imagine that it's like an array where you add genetic material of the extract, of the algae actually, and the plant. I see what transcribes from it, what protein I obtain from that, which is quite different from the logic I mentioned before, that we would say it's a traditional one. Because if I manage to understand what genes are activated by having those components in the formulation, then I will know perfectly the effect I'm going to achieve, and I'm going to reach that function that I want with the biostimulant. So, as I was saying, the genomic effect, here you have an example of what happens with Megafall, but all products have the same approach. Then we're going to see that at the end about what Megafall does and what the study said. But the number of genes it activates is huge, and one of the differential factors has to do with all the things we manage the plant to do naturally in order to stop the effects of stress, and in many aspects, the oxidative stress.
But we will get to go over it towards the end in the concepts. Phenologically, we need to understand what we manage in terms of growth and development of the crop. And at this point, we have lots of prototypes to test, many to see and collect and understand what the prototype can provide. After that, there's testing and in greenhouses and then on the field. But at a prototype level, this is really very big. There's lots of things behind it. Megafall, to understand the mode of action, we said we will carry out all the tests. One of the distinctive factors, one of the hallmarks, is the gene activation of a group of genes that allow me to focus on these two big axes the crop has to fight stress. If you zoom into it, it is to fight oxidative stress and to activate, to trigger growth, to protect cells, because there are components, and it activates genes that activate osmolytes. They protect membranes. You have to think about the fact that there are amino acids, and if you increase the concentration of amino acids, you are somehow shielding the membranes, and you are protecting them from the potential stress that can happen. Then it also increases gene activities, generating molecules that are natural antioxidants that belong to plants, and they activate photosynthesis. So we have the most important factors, and the benefits of this product have to do with overcoming stress, with activating metabolism, both under a stress condition and under a non-stress condition, for machines, for plants to have an escape mechanism and to protect it from thermal stress, from droughts, from haze, from other conditions. So under all these factors, we have been working. There's a lot done in Italy, in Brazil, and in Argentina. We've been working on this for 3 years to understand and quantify how Megafol helps us. And we have very good results. In closing the presentation, we will then move on to questions and comments. I would say that Megafol takes action on 127 genes. There's some that help me overcome stress, and there's others that trigger growth. How is that so? Through increased vitamins, through increase some amino acids. Some are osmolytes, and others are necessary and essential for the rest of the metabolic pathways to generate energy in the cell. And this, of course, has an impact on development and on growth. In connection to stress, in these 127 genes, there is a group that has a direct approach, that of activating those genes to avoid all these types of stress. We didn't see this in so much detail, but of course there is stress because of salinity. This is one of the problems, and it is presented on one of those papers that I've shown you today. One of the big problems that have to do with stress, and it erodes soils as well. So this work with Megafol and this approach is something that is quite distinctive. It's a hallmark and a very good part that this product offers, bearing effects and that are visible in the field. It is a pretty flexible product in connection to use. I had to work in development locally with a team. We did have records on several extensive crops, wheat, sorghum, and barley, and the effects, the biggest ones we've seen by using these products at early stages. Because what we give to the plant, well, if we plan ahead or if we anticipate a stress condition, well, in agriculture per se, we are doing like open pit mining, if I may be allowed to say so, and it's far more difficult to see how serious stress can be and to forecast it. But the logic has to be preventive when it comes to the use of this type of products. This is why this is recommended. And we saw benefits in applications in vegetative stages. When being used at reproductive stages, you end up seeing the performance or the yield, but we collect very good results when we apply it early. And there's also records obtained for fruits and for fresh vegetables. This may not be the focus of attention of my lecture, but megafall will surely start appearing on other crops along time. This is related to what I was saying. This is the positioning. It's quite ample. Why is that so? Because of what we were referring to. It is a flexible product. It is very compatible to apply with pesticides and fungicides. We continue to carry out compatibility tests.
Sabemos que
Portfolios get to change. Stimulants are part of the diversity of decisions that farmers have when mixing with a multiplicity of products. We are working to understand this. But based on what we know so far, it is very compatible. And some results. We had very dissimilar seasons. 22, 23, severe stress because there were droughts, where we did get responses, and 23, 24 was closer to normal. We know that stress did not disappear, but we find positive responses in all the conditions in all seasons. And this speaks very well in a big number of tests that the activation phase is quite efficient and quite truthful. So this is what I wanted to talk about when it comes to megafall. I remain open to your questions and comments. Thank you. We will have a space for questions and answers. The microphone is in the room. So please raise your hand so we can ask questions. That was an excellent presentation, Hernán. Thank you so much. Our colleague here has a question, the first question. Good morning. How are you doing? Thank you so much for this very clear presentation. This is an issue that brings about a number of doubts. You didn't write radiation stress. Is that because you don't consider it? Or is there any special way to see it? It is considered. It is on the first list of stress factors, and maybe towards the end it is on a smaller print. It's included within the stress factors. We see that since it does have this protection effect on cells, megafall helps us protect or take care of crops. In intensive crops where we have more cases of radiation stress, that is where we get to see effects that can be seen by the bare eye. But it is our understanding, based on all the tests carried out on the so many seasons so far, is that there is protection against radiation in extensive crops. When it comes to soybean, this is the crop where it can be seen visually, and also in corn. We see it because that situation is avoided, because we see that the leaf may wind upon itself, and we also see it based on the spots that the sun leave on crops. How long do we have to let by or allow it to pass for us to consider it's been successful? You talked about stress throughout the cycle, but we consider we have not that many chances. We are considering one or two. I want to know how long the product lasts inside in order to administer it the best way. That's a very good point and a very good answer, a very good question. I thank you for that. I was talking about hormones and cycles. We have a given concentration of giberellin or cytoquinine. These are an internal report, in a way, for the plant. And these are the points at which we may even perceive visually on the crop that there is stress. And we will see it because we monitor conditions the crop is going through. So having stated this, if we think about positioning and if we think about how it is applied, the gene activation effect is pretty persistent. These cycles and these tests were carried out in Italy, and part of them were also carried out in Brazil. They let us know, they allow us to know that stress reduction, when you apply once in an early stage, it comes down. And then we recommend to apply on a second stage. Our understanding shows that we don't need to apply more than twice. There's countries, however, even in Brazil, where we apply the biostimulant up to 5 times. But the logic is very different. If we wanted to keep homeostasis at a perfect balance, we need to understand upfront that 2 applications are not enough. But this requires after-the-fact work in order to adapt this management to our conditions, which are different from those of other countries. And this is what we've been doing so far. Thank you, Kako. There is a question here on the front of the room. Good morning, Alfredo Garcia from Alberti, Buenos Aires. In this case that you presented, we can take products from other companies. What comes to mind? Well, you said, let's remember our days in college, cycles. And I was trying to memorize the physiological processes that happen in the plant and the plant response or reaction in view of situations that trigger stress. So my question is, if this product is applied under situations that trigger stress, what about the purpose? Is it to generate a response by the plant that is equivalent to the hormonal physiological effects, or in replacement of these hormonal processes during stress or post-stress? Why is that so? Because if stress is caused by dehiscence, or deficiency lasts, in order for this to translate into growth and development, it should stand in place of the deficiency of the factor that produces stress. How would I work there? I guess you talk about nutrient deficiency. It might be water deficiency, high temperatures, salinity. These are permanent situations. Well, it's more general. With this approach, well, these 127 genes, there is a group that is almost half that relate to or have an approach or the focus on stress, and another one on growth and development. And it comes down with an effect if you apply just once, related to what we were discussing with Kako. They come down progressively, because you were asking what is happening with deficit over time. But they are there. So the group of genes that I activate in surpassing stress, they relate to suppressing or something similar that you were just saying. And the other group of genes, it doesn't have to do with the plant increasing, but not exceeding, but increasing productivity. That's the balance, because if stress condition endures over time, we clearly won't reach the maximum possible productivity, if you follow what I say. I do. And at the same time, if we apply the product under the stress condition, the plant, does it have mechanisms that were dormant that have to be activated to include the product and trigger the response to the product? Well, there is no additional cost on the part of the plant, especially in the group of genes, stress genes. There might be an ATP involvement at a cellular level, but in growth activation, it has a lot to do with increasing amino acid concentrations that the plant produces in and of itself. And some are cofactors to generate vitamins. The plant then uses this as a resource. I would say that if there is a cost, it's very low. This is what I think you were referring to. Additionally to energy cost, whether it is receptive or not. That is to trigger the reverse process. This has more to do with the physical process. Product penetration is excellent in terms of quality, application quality. This is why we say it is pretty flexible. Stress condition of plants, it limits you to applying other type of products. For instance, when we see a very stressed crop, we decide not to apply herbicide or fungicide under very extreme conditions because we know there is a cost to the plant to detoxify. This doesn't happen with Megafol. It never happened. Thank you, Hernán. Let's move on to the next question. That's a very good lecture. In connection to stress, well, it is a very complex issue because unlike the majority of the agricultural practices we have, where we solve specific problems, dealing with stress is not easy. And when it is because of nutritional deficiencies, we should think about solving it by adjusting nutrients. From the point of view of water, well, that's quite complex because when we have terminal stresses that cannot be solved with water, we sometimes move the needle with these products, but we continue to have droughts and nothing can be done. However, modern agriculture today shows we have some agricultural problems that have to be solved, that have to do with stresses because of phytosanitary products, pesticides or herbicides especially, and people are concerned based on what they see. But things that are not seen are also part of a good agronomy. Subclinical phytotoxicity of herbicides takes up many more kilos than people think. A lot is being done in connection to this. So we are thinking a lot about this today. There's a group of professionals that are highly concerned because there's a lot of concerns about herbicide-resistant weeds, and these generate subclinical phytotoxicity and silent losses. This is to speak very bluntly about the whole thing. So we are thinking about this type of product. We are from Uruguay. We take a close look on this product to see whether it plays in our favor. Maybe not for the plant to better withstand stress, but for the plant to recover better. And there's always a question. So this product, is it mixed with the herbicide or is it applied after the herbicide? That's a very good point.
The works we have conducted so far and part of the development of Megafol in other countries indicate that if we have a way of anticipating that stress condition, and in this case we know because it's induced, we don't have to forecast a condition, it's more convenient to apply it beforehand because better effects have been observed. That doesn't mean that there will not be an effect because I know what the practice is and what the logistics is if you apply it at the same time. So the higher benefits are obtained if you apply it in advance. About the herbicides, that is quite complex. Locally in Argentina, we are working on it. I I think that what you're saying is also very true, as the previous gentleman was saying about the nutrient deficit. Perhaps it requires another specific talk about it, specifically for managing that kind of stress. Because going back to herbicides, it's difficult to measure accurately what effect you're having based on how much herbicide you have in the soil. In the latest seasons in Argentina, that has happened. Because of the drought in winter, we have carryovers of herbicides. So you can estimate how much you're losing in crops, but we have to find a correlation and understand and be able to quantify how much it helps us, megafall, for instance. But it's quite complex. These are midterm trials. Next question. Thank you. We ask you, please, to be very brief in your question. I just wanted to know the percentage of algae contained in megafall. I assume it's Aquafil 1, 2? Yes, the base, actually it has extracts. About the percentages, you have most of them is Ascophyllum extract that comes from Ascophyllum nodosum, I would say around 80%. But in that 20%, there's another combination with other algae, which is not Ascophyll. The basis of the product, anyway, it's not comparable actually. In the portfolio, there are other products that are specifically Ascophyllum. Ascophyllum in the large group of algae that we use to obtain the extracts is the most studied, but this comes from a previous work of Balagro. The species were chosen based on their components, which is not Ascophyllum. Great. We have reached the end of our time, so we're going to finish the Q&A here. I'd like to thank you all, those who asked questions, and Hernán for your presentation. So thank you so much. Thank you.